the thing that to appreciate is that current estimates are that there are about 1 million species of parasitoid wasps worldwide.
That is crazy. Hello, everyone, and welcome back to People, Parasites, and Plagues, a podcast aimed at delivering information about the fascinating pathogens among us from the scientists who study them. I am David Peterson.
And I am Kim Klinowski, your host for today's episode. If you thought the movie Alien was horrifying, it's a good thing you're not a caterpillar.
Our guest today is Dr. Mike Strand, H.M. Pulliam Chair in the Department of Entomology. He's also a member of the Center for Tropical and Emerging Global Diseases and a member of the National Academy of Sciences. Mike, welcome to the podcast.
Thank you.
Well, Mike, the first question that we like to ask our guests is about their pathway to science. So how you became interested in science, and you can start whenever you choose to, at as young of an age as you choose.
Well, that'll be pretty easy. When I was pre-college, I was always kind of interested in stomping around in swamps and picking up plants and animals and things like that. So I always had an interest in life science, or at least natural history. And I went to, when I entered as an undergraduate, I was also simultaneously totally clueless about what I really wanted to do. I thought deep down inside I wanted to work on fish on some level because I like to go fishing. So it was really well thought out. What happened was I worked for a geneticist as a sophomore, karyotyping fish, which I thought was kind of my calling, and I learned it wasn't exactly. Some of it had to do with lab dynamics. Some of it had to do with just not really catching on to that. And the following year, I took a general course in insect biology, and I really liked it. I don't know why, but it resonated with me. And that year, I also got a really prestigious lab dishwashing job in entomology.
We've all been there.
Yes, we have, and
Yes.
they're all equally prestigious. But in the process of washing dishes, I was given a couple of opportunities to do a few simple experiments working with another person. And it was trying to identify a pheromone in fire ants, of all things, and it regulates their foraging behavior. So I got to do a lot of bioassays, and I was sort of a half biology chemistry major. And so there was a lot of trying to purify this molecule. And I'd never had any experience like this before, but I was totally taken with it. Absolutely cool. And kind of a welcoming lab environment. And I just learned that I had a real affection for doing simple experiments, and that I had a thing for being able to do mindless repetitive work, because there was a lot of mindless repetitive work with trying to purify a pheromone. That's pretty much what happened. And this lab also worked on parasitic insects. So that was my first exposure to that side of things. And I thought I was oblivious to these things existing. I thought they were just the craziest, wild, wonderful animals I'd ever seen. And that set the tone. And ironically, I ended up staying in that lab as a PhD student. So I kind of broke the first cardinal rule of, diversifying your portfolio and getting new experiences. I was provincial to a fault and didn't do any of those things. And I ended up staying there and finishing my PhD and then postdoc and all that sort of stuff afterwards. But it was all a kind of undergraduate experience, which is so common for so many scientists that I've met.
Yeah, something that really kind of lights your fire and helps point the way.
Yeah, it's kind of a two-way street, because some of it has to do with creating a nurturing environment, but some of it has also to do just with the innate tendencies of people that sometimes they don't know are really there. So you have many people who have that experience, and they would have just thought, please kill me, after having pulled abdomens of fire ants all day and stuff like that. And yet for another person, it's kind of magical and it's really interesting and fun. So it's sort of a little bit of both, of self-discovery in younger people and the right environment to do some self-discovering. And sometimes those things match and sometimes they don't.
Yeah.
And it's exciting when it happens,
yeah,
when you're on the other side, for
absolutely.
sure.
So, Mike, what are the fundamental questions you pursue in your current research?
Well, I sort of see myself as kind of a jack of all trades, master of none kind of biologist, and have worked on a lot of different kinds of things. So I think the main things are that I've always really been interested since my undergraduate days in parasitic organisms and in associations with microbes. And so I've worked on a lot of different study systems, but they've recurringly kind of revolved around multi-organismal interactions. I started out as a more kind of behaviorally oriented, almost ecologically oriented person, and realized, I think, that I wasn't actually that good at that. There were just lots of people who had more talent than I did, and maybe a deeper affection for it. And I slowly kind of started drifting into more sort of suborganismal themes and mechanisms and a little more biochemistry, molecular biology kinds of things, but always with a little bit of an organismal eye because I kind of came from an organismal background. And so I think that everything I've worked on is redundantly having to do with insects and has but many different types of insects and a lot of different kind of microbial insect associations, and just let kind of projects lead me as opposed to the other way around. So you sort of observe something, it seems kind of interesting, and sometimes those bear out and have some traction, and sometimes they don't. But I'm not a kind of one-horse Charlie person, really, in terms of the lab. Always have kind of juggled several balls. And I think sometimes what comes from that is that there's strength in the diversity, but there might be weakness in terms of depth. And some people build careers around developing deep expertise, but maybe on a relatively narrow front. And other people contribute in a more breadth-oriented way. I guess I kind of put myself in the latter category.
Okay.
Mm-hmm. Now, Mike, I mean, clearly you've articulated that you work on lots of different things, but one of the things that I think we want to just focus on today is the parasitoid wasps. Mm-hmm. So you mentioned you were into insects early on. Were you studying those wasps early on too? Oh, yeah. You were? Okay.
Yeah, that lab that was trying to purify that trail pheromone with ants, I did a senior thesis project on a parasitoid wasp, and it had to do with how factors that regulated foraging, that is, how they locate host organisms. So these insects, as you mentioned, Alien as the movie, all borrowed from the biology of these kinds of insects. So they lay their eggs in or on other insects where their progeny develop. So very much like in the Alien movie, what's being parasitized are other insects instead of people. But the way that the offspring of these parasitoid wasps develop is very much like in the movie. And they, more often than not, develop internally. And then when they complete their immature life cycle, they emerge from their host, which is pretty dramatic and kind of gruesome. Mm-hmm. But they also have evolved an amazing repertoire of strategies for manipulating the biology of host organisms to the purpose of facilitating the development of their offspring.
So I think we're going to talk about that in a lot more detail in a second, but before we start that, the only thing that comes to my mind, and is this the same organism, is when you have the tomato hornworms and they have all the little cocoons coming off of them. That's a parasitoid?
It's a parasitoid wasp. Okay. One thing that people generally, first, these insects are mostly very small, so they're not real apparent. Tobacco hornworms are pretty apparent.
Oh, yeah, very, and especially if you're growing your tomatoes.
Exactly.
Yes.
So you have these huge caterpillars. But the thing that to appreciate is that current estimates are that there are about 1 million species of parasitoid wasps worldwide.
That is crazy.
So that means that they belong to the order Hymenoptera, wasp, bees, and ants. So things like honeybees and yellow jackets and stuff are in the same group. But about 80% of the group are parasitoid wasps. They're not ants, they're not honeybees, and they're not those sorts of things. And if there's 1 million species of them, it means that they're the largest group of insects on Earth. And so since insects are about 75% of all described species of animals on the planet, it means a substantial proportion of all the animals on the planet are parasitoid wasps. There are genera of parasitoid wasps that comfortably exceed the total number of known species of mammals, which is 4,500 to 5,000 species, which is trivial in comparison to 1 million species of these organisms. So this kind of biological habit has obviously been super successful. And they impact the planet hugely because they are such an important mortality agent for insects, which are hugely impacting the planet because of all the pollination activity and decomposition activity, stream ecology. There's no ecosystem that could be what it is without insects in it. And then virtually all of those insects are being parasitized by parasitoid wasps, who are all very, very specialized to develop in particular species of insects or other arthropods. So right under your nose are just an untold treasure trove of diversity that's pretty amazing.
You know, in beginning parasitology class, you tell students that most organisms, certainly higher eukaryotes, have multiple potential parasites. And I guess it's the same thing with insects then.
Yeah,
so there's
absolutely.
That's why there's so many parasitoid wasps,
right?
Because
they specialized biology drives speciation events, and so you end up with these guilds of parasitoids that are using hosts. So almost every host is being parasitized by multiple parasitoid wasps, which are using different life stages, and they keep splitting the resource into finer lines so that they all coexist, but it's also an arms race of driving increased specialization recurringly again and again and again.
So one of the more interesting findings you've reported is the role of viruses carried by the wasp that are injected into the host. And is it the actual virus particles that are injected?
Yes, it is. It's a kind of a complicated story, but also elegant, I think. If you don't mind me just sort of providing a little teeny bit of background.
Absolutely.
We have 1 million species, give or take a few, parasitoid wasps. And we don't really know, but maybe 100,000 species or so have evolved associations with viruses like this. And that's just a back of the envelope estimate of how many, but the point is a lot. The way that these associations have come to be, which we've only learned over the last 10 or 15 years really, is that there are viruses that infect insects just like there are viruses that infect all eukaryotes. What happened is that there's particular lineages of viruses that integrated themselves into the germline of certain lineages of parasitoid wasps. So the virus became part of the genome of the wasp. And rather than being able to produce a virus that replicates itself and can then disseminate and infect other organisms, this integration event got manipulated by these wasps. And so they have taken the genomes of wasps and they've changed them a little bit. And so now these ancestral viruses, we refer to them as being domesticated. And they, in effect, are only capable of replicating in the reproductive tract of female parasitoid wasps. And they produce a virion, they produce a particle that packages, in some of the lineages, packages DNA, because these are the ancestor with a DNA, double-stranded DNA virus. But what gets packaged into the particles is the machinery of the ancestral virus makes the particle. But now they package DNAs that are only partial, they're substantially not really viral anymore. Mm-hmm. So they've created what we call a gene transfer agent. So they have used the machinery of viruses to make virus particles, but to package novel genes into the virion, as you would with a gene therapy project mm-hmm. of, let's say, using viruses to introduce DNA into another organism, as we do in medicine, with certain types of viruses. So the parasitoids have essentially evolved the same kind of strategy, and they use these particles to transfer genes from themselves into the genomes of the hosts they parasitize so that they can express genes that change the biology of the hosts in a way so that their progeny can develop properly. So they immunosuppress hosts, they change the way they grow and develop, they turn them into zombies, they behaviorally change them. So they have this large inventory of genes that they have co-opted the ancestral virus to deliver into the host. So what has happened is it's an elegant example of repurposing of a virus, changing the genome of a wasp, making a new entity, and the wasp uses that to be able to parasitize its hosts, other organisms. It's pretty sexy.
I mean, I don't even know what to say. I'm so flabbergasted.
Only evolution can ultimately be so elegant at times.
Exactly. And I wonder if we can just talk about how much of this is chance, right? Because at some point there had to be, I'm assuming, an integration in the right place for this to be passed reproductively in some way.
So that's a very good point that you make, Kim, because the viruses in the wasps that we study the most evolve from a group of viruses that are in a family called Nudiviridae. And that's an insect-specific double-stranded DNA virus that's pervasive all over the world, infects all kinds of different insects. Where people listening to this podcast might be able to relate to nudie-viruses, because probably most people haven't heard of them, is there's another group of viruses called baculoviruses. And baculoviruses are used extensively in biotechnology and things like that as an expression vector mainly.
Yeah.
Used in vaccine production and all kinds of different things. And we know from increasing inventory of genomes to survey that there have been many nudie-virus integration events into all kinds of different insects. Just like a lot of viruses, fragments of viruses are in vertebrates and plants and things like that. And most of them, under neutral selection, they mostly just die out. So you just end up with bits and pieces, but they're not functional anymore and they decay, and that's the end of them. In a few rare circumstances, they end up integrating into a location and in a way that they've been able to persist, and for some evolutionary trajectory to go like I'm describing for these parasitoid wasps. And so we know of many hundreds of insects that have nudie-virus fragments in them, in their genomes, but they didn't evolve into...
They didn't share a common ancestor. This was individual
so there have been many, many integration events of nudie-viruses into different types of insects, because we can find the remnant bodies of dead nudie-viruses in all kinds of insect genomes. But in these parasitoid wasps, it created the right selective environment, combined with we don't really fully understand why these succeeded into this kind of association, but it is a rare event. The point is exactly what you said. It's not like this profligately happens every time there's an integration event. It's the opposite. Most of the time, it doesn't happen. We do not understand why it succeeded in the cases it did, but we know of currently of 3 examples of different nudie-viruses that ended up producing independently different types of particles, all in different lineages of parasitoid wasps. So this has occurred more than one time, but the types of particles that end up being produced are not the same. So I describe the ones we work with mainly, which are really transferring genes into hosts for the benefit of their progeny. We know of other cases where functional particles are being produced, but there's no nucleic acid in them at all. And they instead package proteins.
Oh.
Okay.
And they function kind of like microvesicles or something. So they end up injecting the particles into the host insect, and the particles invade host cells, but they're delivering a protein payload instead of a DNA payload.
Okay, and I assume the proteins probably affect gene expression in the cells, or?
We honestly know relatively little about what exactly they do. We know that by homology, there's a real good, we know what the particles do. So the particles protect the wasp's eggs from the immune system of the host insect. So the host insect otherwise recognizes the wasp's eggs, and immune cells attack the egg and they kill it. When these particles are present, they don't do that. And we know that some of it has to do with they interfere with the ability for the cells to be able to, in insect immunology, a common response is what we call encapsulations, where immune cells bind to the foreign target. It's a little bit like the way a macrophage would function, and you end up walling off the target from the rest of the body. They secrete all kinds of cytotoxic molecules into the capsule and they kill it. When these particles are present that have from the lineage of nudibviruses that didn't package any DNA in the particles anymore, instead they're packaging proteins, this encapsulation response doesn't happen. And we know that among the dominant proteins that are packaged by these particles are ROGAP type proteins and RATGAP type proteins, which are GTPase inhibitors or modulators. And these are famous for their affecting the motility and cytoskeletal functions of all kinds of different cells. And so ROGAPs alter and manipulate the way cells can move or stick, things like that. So the implication would be that they've co-opted wasp ROGAPs, get packaged into a particle that comes from viral machinery, and they can deliver these kinds of ROGAP molecules into immune cells to disable normal function so that their eggs don't get encapsulated. For the ones that we work with mainly, where they are packaging genes, they package a variety of genes that we do know that they interfere with encapsulation, but just by a different mechanism. So they, for example, one of the things they package are NF-kappa B type molecules that interfere with NF-kappa B transcription factors in the cell, which regulate, innate immunity. It's like linchpin signaling. And the NF-kappa B molecules genes that are packaged into these particles have been truncated. So they only have the binding domain of the NF-kappa B. So they're packaging I-kappa Bs, the negative regulators of NF-kappa Bs. And all of the regulatory machinery of the I-kappa B has been kind of culled off, and you're just left with the binding. And when they bind to an NF-kappa B, the NF-kappa Bs can't translocate into the nucleus and activate all these innate immune signaling pathways. So they're preventing the immune cells of the host from functioning normally by flooding them with these I-kappa B inhibitors, and everything goes wrong. And they also introduce other kinds of genes that interfere with other processes as well. So that's completely different from using ROGAPs. But in a sense, or putting proteins in a particle instead of putting genes in a particle. And it just kind of tells you that the evolutionary trajectory in these different lineages are not the same. So they borrowed from nudibviruses to make particles, but then how they use them and what they deliver to host insects is very different in different lineages.
This is reminding me of kind of a heist movie where the crooks gather outside and somebody goes to the alarm panel and they disable that, and someone else gets inside and then they shut down the cameras in the system, then they steal everything.
Well, it's very true. I mean, it's one of these things that we learn in a lot of biological systems, that they're multifactorial and there are many targets that are kind of candidates for manipulating a process. It would be a lot of cancer therapies are really or if you were talking about trying to control malaria parasite development, you're looking for drugs, and these drugs could idiosyncratically affect many different types of targets, and the endpoint might be similar, but the route of getting there might not be the same. And in a sense, these parasitoid wasps in these different lineages have kind of come to different solutions to a problem independently.
So do you have a sense over evolutionary time of when wasps acquired these viruses and I guess also started using them in this way?
In some cases, yes. So the ones that we study the most, we know that all of the wasps we estimate there are about 50,000 species of wasps that have what we call brachoviruses. So these are the lineage of they're from the nudibvirus ancestor, and they produce the particles that put genes into the particle and NF-kappa B inhibitors and things like that. This 50,000 species are monophyletic. That is, they all come from the same ancestral wasp. And we know we didn't do this work, but systematists working in this area estimate that the common ancestor of these brachovirus-carrying wasps was originally infected about 100 million years ago. So that's a long time. And so the radiation of this group is really ancient, and they all maintain this machinery, but there are a lot of differences among these 50,000 species because they've ended up on their own eventually, not exactly the same evolutionary trajectories. But we know absolutely that they come from a single common ancestor. We know that common ancestor was about 100 million years ago.
Okay.
We know of two other lineages that because of the way genes of the virus are organized in the genomes of wasps, we know they're more recent. So in the one we work with that's 100 million years old, we just are finishing a paper, and this is work that I do with my closest collaborator on this work is Galen Burke, and she's just a fantastic scientist. And we've worked together for many years now. We have this chromosome level assembly for the main species we work with, map where all these genes are. And the genes of the virus are on every chromosome of the wasp. So the genome, one thing I didn't say is that the genome of the virus is no longer intact in the wasp. It's over 100 million years. It's ended up the recombination and all kinds of things, crossover events and everything. All the genes are just all over the place, but they still speak to each other. So they can still, the machinery to be able to make a particle is all in there. It's just not like it is in a nudibvirus genome. It's all over the wasp, but they all have the right regulatory elements and the like that they communicate, just like eukaryotic genes can be all over the place and do make something. It's the same thing. So they can make particles, but all the genes that go into making these virions are on all the chromosomes of the wasp. And so it tells us this is an especially fragmented, we refer to it as a viral genome, but it doesn't look like a viral genome anymore. And it's because it's really, really old. And so there's been a lot of movement of all the parts. Whereas in some of the more recent ones, a lot of the blocks of genes are still in tandem array with each other. They're very close to each other. So the genome's more compact and more like the ancestral genome, but they're not as ancient as the one we work with.
And the endogenous viruses are only active and transcribed in the female reproductive tract. So
correct.
As far as you know, it doesn't play a role in male wasps?
Not at all, or in any other tissue. So among the things that we don't understand is the process of making particles is temporally regulated. So it only happens when the wasp is transitioning to becoming an adult. It's sex specific. It only happens in females. And these particles are only produced in a specialized type of cell that we call calyx cells that are part of the reproductive tract of the female. They reside in proximity to the ovary. They're part of the ovary, really. And it's only in those calyx cells. So it's integrated into the germline. So every cell has all these genes, but these genes are silent everywhere but in calyx cells. And at this very particular time, in females, replication in these calyx cells occurs, and you just make billions of virus particles in there. And she just ends up with a reproductive tract full of virus particles, but they're not capable of replicating because they've been specialized to package all these virulence genes. So all the machinery to make particles are there, but the particles that are made are not capable of replicating themselves because they've been changed into becoming a vehicle for delivering other kinds of genes into host insects.
I just want to ask a question for clarity. Maybe it's obvious, but I just want to make sure that we're clear also for the audience. What is being packaged? Did that come from the virus genome or from the host genome as part of the packaging process?
It's a combination, as best we understand it. The DNAs that are packaged into these particles are also integrated into the genome of the wasp, and they're amplified, just like a viral genome gets amplified to be able to be packaged into a virus particle. So nudibvirus genomes are circular double-stranded DNAs. They're not integrated, but they replicate themselves, and then they get put into virions as they're being made, just like most viruses. These guys, the regions that are going to get packaged into these particles, are integrated into the genome of the wasp. They're amplified, and they're circularized, and they're put into the particle. So they kind of look like nudibvirus virions. But what's on the DNAs doesn't look like nudibvirus virions at all. And all of the machinery to make the particles, none of those genes are on these DNAs. They're somewhere else in the genome. So the particles are being made by viral genes, no doubt about it. But these domains of DNA that are amplified and are going to be processed into circular DNAs that get put into the particles, the flanking regions of these DNAs probably have antecedents to the virus. We're not absolutely sure about this, but there are signatures that they do because viral genes act on them to amplify them and process them.
Right.
We know that. So we know some of the replication machinery is there to work on these things. But everything that's in these DNAs is not viral. We know that some of them are wasp genes because there are paralogs of, some of these are gene families. You know that there are other genes in the genome and they've just been moved into these DNA regions. Some of the genes, we really can't tell where the heck they came from. They don't seem to be wasps. They don't seem to be virus. We're not really sure where they came from. And a few things might be from the virus ancestor. There are some things that show some signatures that they might be, but we're not sure. So it just means that a lot of different types of genes have gotten recruited into these DNA domains. But it's sometimes a little confusing to separate what's a viral gene and what's not a viral gene, even within regular viruses. So like viruses that have big genomes, like nudibviruses or baculoviruses, there are genes that we all can agree on as being viral genes because all the viruses have them. So there can be thousands of isolates of baculoviruses, and they all have these genes. And they regulate mainly replicating the genome. They regulate making particles, that sort of thing. We call these core genes. And almost all of the nudibvirus and core genes they share substantially with baculoviruses are present in these wasps too. Okay, so the core gene machinery is there. But in regular baculoviruses and nudibviruses, there are other genes that are specific to a particular genus, or they might be in some genera, but not all genera of the family. And then there are hundreds of genes that are unique to a single isolate of virus because they horizontally acquire genes from all kinds of places. So you've got core genes that are universally, we can identify as those are baculovirus genes. But then there are these kind of wanky genes in there that they kind of picked up from who knows where. And they're unquestionably in the virus, so they are viral genes in a sense, but they're not ancient viral genes because different isolates have substantial numbers of genes that are very unique. It'd be, like why this, a bacterium like E. coli causes somebody to get massively sick because it's oddly picked up toxins or virulence genes that make it a different variant of E. coli versus another one which we're growing, in the lab routinely, and it won't make you sick. Viruses do the same thing. So it just means to say that even in these blocks of genes, there are genes. A lot of viruses have eukaryotic genes in them. Right. So it's very, very difficult to sometimes separate what's virus and what isn't.
So did these viruses affect the host at all?
The host that they...
the wasp. Did they ever affect the wasp?
That's a great question. And since everybody's infected and everybody's a carrier and we can't cure them because they're all integrated into the genome and there's hundreds of genes and they're all over the place, we can't really make a wasp that doesn't have these genes. But we also, if you compare these kinds of wasps to wasps, parasitoid lineages that do not have any of these viruses, they show no unique features at all. It's not like they live longer or shorter or they're bigger or they're smaller. There's no phenotype. And then virtually all of these genes are silent, except in these very specialized cells. Right. So we think on balance, they do not.
So earlier you mentioned how important insects are to a healthy ecosystem. And I'm wondering about the role that parasitoid wasps play. Do they tend to favor what we might think of as detrimental insects as hosts, or do they also, are they equal opportunity and they'll lay eggs in so-called beneficial insects?
So they given 1 million species, and since very few insects are not parasitized by something, they're equal opportunity destructors. But what led to people studying these animals is because many species do parasitize economically important insects in forestry and agriculture. And in many cases, they play a substantial role in reducing the population density of a variety of organisms that otherwise would be pest insects. So in most of our pest species in agriculture and forestry are actually introduced. So they came from somewhere else in the world. And a lot of times, people don't fully understand why introduced species might become a pest. But one of the schools of thought is they become pests because the organisms that maintain them at densities that are too low to achieve pest status, because there are not enough of them to defoliate your orange tree. Mm-hmm. It's because there are other organisms that are maintaining those populations at lower densities. And the insects that are substantially doing this are parasitoid wasps. So for centuries, humans have known that they could use these kinds of organisms to control pest species. So biological control in agriculture and forestry is just a generic term for using parasitoids or predators or pathogens as control agents for pest species. Classical biocontrol is really the idea of taking an introduced pest and then identifying what it is, which is sometimes not easy, and then going to the native range of the pest organism, where a lot of times it's rare as hen's teeth. You just can't find these things. They're not common at all. They're definitely not a pest. And people seek to identify predators and parasitoid wasps that are using them in their native range, screen them for specificity, and people are very, very careful about this because the idea is to introduce them into the introduced range of the pest organism with the hope that they will establish themselves and achieve densities that they control the pest. This has worked famously well many, many times, and it's a little bit like vaccination in the sense that if it's successful, it's relatively low cost and it's permanent. Forever.
You're just reestablishing
what happened in the natural habitat of the pest species. The challenge in biocontrol is that a lot of times it's not very predictive. It's very difficult to know what will establish, whether it will have the desired effect that you want. A lot of times you release them and that's it. You can't recover them again. They didn't establish. Sometimes they're there, but they don't have sufficient impact on the pest to change its pest status. And then other times it's a home run and it just works awesomely well. So cooperatives and growers have benefited enormously. We have biocontrol programs all over the country and all over the world that have been spectacular successes. And it means that we don't have to use insecticides or other means of control. And it's, by my perspective, there's cost benefits with any sort of pest management strategy. But to me, there's definitely more pluses than minuses to the approach. And that is where really a lot of the studies of parasitoid wasps begin. It's because it's studying species that are parasitizing pest species. And they were collected and they were worked on initially because of their potential as a control agent without genetically manipulating them or understanding how viruses work, or that the viruses are actually responsible for why they are an effective biocontrol agent. It's just trying to find something that would succeed without understanding the mechanism mechanisms why it succeeds. But that's really where the study of parasitoid wasps began, and that's usually what drives a lot of the organisms that then get worked on in a more basic science kind of way to sort of retrospectively understand why are they good biocontrol agents, or why might they be a crummy biocontrol agent.
So, Mike, when you're not thinking about wasps and mosquitoes for that matter, what are you doing in your free time?
I think my favorite things to do in free time is for about the last 30 years or so, I got really interested in horticulture, and I've become a kind of a plant geek. I've spent a lot of time kind of on garden sort of stuff, totally ornamental, very esoteric, but it's really just working with different kinds of plants that I think are really cool. My wife and I, we bought a cabin about 15 years ago on the Georgia-North Carolina border on near the Nantahala Wilderness. And so we've always had a thing for Southern Appalachia, kind of walking in the mountains and doing things that are related to that part of the country. It's one of the reasons I moved here. Was because both of us really liked Southern Appalachia. And Athens just was close by, and that was good.
Perfect.
Yeah,
Perfect. So
location.
that's kind of what we do in our free time.
Oh, I love that. So our guest today is Dr. Mike Strand, H.M. Pulliam Chair in the Department of Entomology. He is also a member of the Center for Tropical and Emerging Global Diseases and a member of the National Academy of Sciences. Mike, thanks for being with us today.
Oh, thanks for asking the questions.
Links to additional information about Dr. Strand's research can be found on our Instagram account at PPP Podcast UGA.
As always, we appreciate you tuning in today, and if this is your first time listening to the People, Parasites, and Plagues podcast, we encourage you to check out the other exciting episodes launched last season. Please rate the podcast, and if you have any questions or suggestions for future episodes, reach out to us via email at ppp@uga.edu. This podcast is brought to you by the Faculty of Infectious Diseases and the Grady College of Journalism at the University of Georgia. It is supported by the University of Georgia through the Office of Research, the UGA Graduate School, and the College of Veterinary Medicine. Special thanks to our production assistant, Justin Nemitz, for research, editing, scheduling, and keeping us on track.